Everyone, welcome to Assis Biology. In this video, we are going to discuss about spermatogenesis, and this is the third part from the chapter Human Reproduction. I am Assis Kumar Das, student of MSc Animal Biology, University of Hyderabad. And if you are new to this channel, please subscribe the channel and press the bell icon for getting more updates. Now, let's get started. So first of all, what we will learn today, we will see what is spermatogenesis, we will see what are the cells that are involved in the process, then we will see what is spermiogenesis, what is spermiation, and we will see many more things. So what that is spermatogenesis, then the spermiogenesis and the spermiation, they are means appearing similar, but they are having large differences, huge differences in their meaning. So we will also correlate these differences. So first of all, what is spermatogenesis? So spermatogenesis is the process of formation of mature motile spermatozoa. That is, for the fertilization we require male gamete and female gamete. So the process of the mature male gamete inside the testis from the spermatogonia is called the spermatogenesis, and it starts at puberty, that is after the attainment of the adolescence. Spermatogonia are the germinal cell, that is the cells that are having ability to give rise to form gametes or give rise to gametes. So our body is composed of two kinds of cells, that is somatic cell and germinal cell. Somatic cell are our body cell, and the germinal cell are the cell that are having ability to form gametes. So these are the germinal cell on the wall of the, that is the inner wall of the seminiferous tubule. So what is seminiferous tubule, what is the means the structure, etc., we have already discussed in the male reproductive system, and if you have not seen the video, I will mention the link in the description box so that you can watch the video. Then the spermatogonia are the progenitor or the precursor for the spermatozoa development, because they initiate the process of the spermatogenesis. And some of the spermatogonia, how this process is happening, some of the spermatogonia which accumulate the rich of food, and the size will increases, they will become double, and they will become the primary spermatocyte. So here you can see in the diagram the spermatogonia is becoming the primary spermatocyte. However, here the size is almost same, but they will increase in size. Then you can see the secondary spermatocyte, you can see the spermatids, and you can see the sperm cell. Here the secondary spermatocyte is written 2n, but this is a mistake here, sorry for that, I apologize for the mistake. It will be haploid, that is the secondary spermatocyte will be n. Now you see the wall of the seminiferous tubule. So the testis is here, this is the seminiferous tubule, and this is the wall of the seminiferous tubule. You can see here, this is the cross section. So here you can see the spermatogonium, these are diploid cell, the primary spermatocyte that are the haploid, sorry, these are also diploid cell, they are formed after the increase in size by the accumulation of food material. You can see the secondary spermatocyte, these are haploid. You can see the spermatid, these are also haploid. So what happens, you can see here the primordial germ cell, then mitotic division is there, becoming primary spermatocyte. Then the fast meiotic division, they will become the secondary spermatocyte. So primary spermatocyte are diploid, and the secondary spermatocyte are haploid. Then the secondary spermatocyte will undergo the meiosis 2, and they will become the spermatids, these are also haploid cell. So we will see all this process in detail. Then the spermatid will form the sperm cell or the mature motile spermatozoa. Here you can see the Sertoli cell, this is the Sertoli cell, they are also called as nurse cell because they provide nourishment to the developing sperm cells. Now each primary spermatocyte undergo meiosis 1, and it will form two secondary spermatocyte, and each secondary spermatocyte will undergo meiosis 2, that is the second phase of the meiosis, and form two spermatids. So from one diploid primary spermatocyte cell there will be four spermatid, 2 from each spermatocyte. So there are total 4 spermatid formed from the spermatocyte. So one diploid primary spermatocyte will lead to formation of 4 haploid spermatids. Now spermiogenesis. The spermiogenesis is the process of the transformation, that is from the spermatid into the spermatozoa. So here what happens, to become mature and to become motile, the spermatid will lose some of its mass, and it will become means elongated, it will means form a flagella, the shape change in shape in the nucleus, then the change in shape also of the whole cell will lead to formation of the head, midpiece, and the tail. So I put this diagram only to make you aware about what are the steps. You no need to remember these things, but you can see here the spermatid nucleus and the sperm cell, that is the spermatozoa nucleus. You can analyze the differences in the size, in the shape, etc. The tail is there in the spermatozoa but not in the spermatids. So spermatogenesis is the process that is the transformation from the spermatid to the spermatozoa. Then the spermiation. So after the spermatozoa become completely formed, they must leave the, that is your seminiferous tubule. So they become embedded in the Sertoli cell and they are released from the seminiferous tubule by the process of spermiation. Then they will move towards the epididymis. From there it testis through the vas efferentia. Now how the spermatogenesis is controlled. The spermatogenesis as well as the steroidogenesis, that is the synthesis of the steroid hormone in the testis, is under the control of the hypothalamus and the anterior pituitary. So as you know, the hypothalamus is the master regulator of the body. It stimulates the anterior pituitary by secreting GnRH. So GnRH means gonadotropin releasing hormone. So as the name suggests, the GnRH will stimulate the release of gonadotropin, and the gonadotropin are, that is your FSH and LH, they are secreted from the anterior pituitary. So why they are called as gonadotropin? Because they stimulate the gonad to release other hormones and other chemical substances. So the FSH and LH are the gonadotropins that are released from the anterior pituitary under the influence of the hypothalamus. So after FSH and LH are secreted from the that is the anterior pituitary, they will stimulate the Sertoli cell. That is the FSH will stimulate the Sertoli cell to release inhibin and other factors that help in the process of spermatogenesis. And the LH, that is the LH is also called as ICSH, that is your interstitial cell stimulating hormone, because it act on the Leydig cell, that is the interstitial cell that is present in between the seminiferous tubule. So the Leydig cell are stimulated by the LH, that is your luteinizing hormone, or ICSH, interstitial cell stimulating hormone, to secrete the testosterone. Testosterone is an androgen, that is a male sex hormone. So here what happens, you can see in the diagram here, the FSH, LH, then you can see the Sertoli cell, you can see the Leydig cell, and the testosterone stimulates the process of spermatogenesis. So ICSH, that is your LH, act on the interstitial cell and stimulate synthesis of testosterone, that is androgen, and the FSH act on Sertoli cell and stimulate the Sertoli cell to release some factor that stimulate spermatogenesis, and also it releases inhibin. So the negative feedback mechanism, that is, what is negative feedback mechanism? So negative feedback mechanism is the mechanism, or it is also called a negative feedback loop, that a substances will repress the secretion or the production of another substances that is responsible for its own production. So here you can see the testosterone is released by the influence of LH on the Leydig cell, and the LH is released by the influence of GnRH on the anterior pituitary, on GnRH on the anterior pituitary. Okay, so the testosterone inhibits GnRH secretion, but the GnRH is responsible for the testosterone synthesis, but testosterone is inhibiting the synthesis of GnRH. That's why this is a negative feedback mechanism. Here you can see the testosterone is preventing the secretion from the anterior pituitary and also from the hypothalamus. Here the inhibin also, as the name suggests, it is inhibiting the secretion from the anterior pituitary. Now the inhibin also inhibits the FSH secretion from the anterior pituitary. Now what is the structure of spermatozoa? It is having head, neck, middle piece and tail. Here we will see all these things: head, neck, middle piece and tail. Then the head is the elongated part. It is compact and haploid nucleus, that is with compacted DNA, that is the paternal set of chromosome is retained inside the nucleus of the, or inside the, in the head of the sperm, that is containing the nucleus. Acrosome is an anterior cap-like structure. Here you can see this is the anterior cap-like structure, acrosome, cap-like structure, and that contains several enzymes to dissolve the membrane. So the egg is covered with a number of membrane, and it must be clipped or it must be dissolved by the acrosome for the fertilization. So for example, one of the acrosomal enzyme, that is a hyaluronidase. Then the neck contain two centriole, proximal and distal centriole. Here you will see this is the proximal centriole above and distal centriole is below. So the two centriole, the proximal is necessary for the fast cleavage division of the zygote. So for the spindle fiber formation, the centriole is required. Then the distal centriole is attached to the tail filament. Here the tail filament is containing axial filament of the tail. So now the midpiece is containing a number of mitochondria. They are spirally arranged, the mitochondria, and it will help to provide the energy for the motility of sperm. And the tail is a axial filament covered by plasma membrane. It is consisting of the flagella, that is the axial filament of the sperm, and it helps in movement towards the male gamete. So the movement of the flagella will help in motility towards the female gamete, during the fertilization. and also the tail help in the direction, means the direction of the movement towards the egg or ovum. So this is the end of the video. Thank you for watching.